Okay, for the last part, very quickly, I would like to show you what we are currently doing. It's actually not so much about the antenna effect, but about playing with optical forces. So if you have light and it hits some material, it actually induces some forces on that material. This is very old. Johannes Kepler explained in that way the comet's tail formation, by that light has pressure with it. And the Maxwell's theory you can find, and Lebedev did the first experiments. And so if you focus down a laser beam, two things can happen. And that depends on the wavelength of the light you take. In one case, if one focuses down a laser like that, it acts like, which is called the gradient force, that these particles want to stay into the focus of that laser beam. So the light is coming from above, the gold nanopart or the particles would just collect here in the focus. At other frequencies, one gets a scattering force, which actually wants to push that particle through the focus. So by changing the wavelengths, one should be able to do both. So this is kind of grabbing it like with a tweezer, but with a light tweezer. And I can show you here the silver nanoparticles actually in an aqueous solution, that if we focus that down, then one can grab these particles at some point in the focus. There's one trap now. And it still moves. It's important for the next view graph. It shows some Brownian motion in that trap. So the second comes actually. They interact and change the color. There's a lot to learn here. The third comes, and so on. And at some point there are too many in there and it breaks down. This was actually also already done also by other groups. What was interesting us, this was this Brownian motion in that trap. And whether we could use that for detecting external disturbances, like fluidic motion or acoustic signals. And that's what we published quite recently, that indeed if one looks at that motion here, one can measure whether there is an external disturbance coming or not. And that's what we called the nano ear. And we are currently, this is, for example, if one has an oscillating tip, one can see that the Brownian motion changes a lot when next to this trap there is this oscillating needle. Currently we are doing experiments with bacteria. So we trap also bacteria next to that nano ear and let that bacteria oscillate with his tail. And we can, I just saw that a few days ago, indeed detect that motion of these bacteria by looking at the Brownian motion of the nearby gold nanoparticle. Okay, but what was, on the other hand, this was done, these tweezer experiments are done in very different ways. What was more interesting for us actually was the scattering force, that we can push particles to the front. And this we tried in a way that we wanted to do that or use that for printing nanoparticles down on a surface. So because if you have a pushing force, one should be able to take a nanoparticle close to a surface in water and push it with light onto the surface and write with it. And that this is possible, I can show you here. So we look through a microscope onto the surface, which you can't see. There are all gold nanoparticles swimming around. And then we switch on the laser, focus it close to the substrate, and then move the substrate. And then one can, sorry, that was the wrong button. And then one can indeed write down things by moving and shooting with the laser one particle after the other onto the surface. And this ends up not in NIM but in SENS, which is the Munich Center for Nanoscience. One can write things like that. We can also, this is a serial printing. We then try to split up the laser in many beams, which can be easily done, and try to do some parallel printing. And this is the next movie here. So if you do that with lots of laser beams, one can produce an array of gold nanoparticles like here and print them all down like an array, and without any lithography, just with laser light. I have this here. Okay, and then we can make different structures, like this simple array. We can put 5 arrays together. We can also use silver. We can make smileys, and we can also make all kinds of structures, which is, for specific applications, nice to have, because you don't need lithography for that, and the accuracy of that method is about 30-40 nanometers, which is for light methodologies not too bad. Just to show you that, this light pressure can also be used, and is also discussed at the moment a lot, to push particles or compounds with particles into cells. And here I show you an experiment, not a cell, but it's a so-called vesicle. But with the laser, we could indeed push the particle through the vesicle membrane into the interior, and show that in principle it should be possible, that might be important for drug delivery things, to push that particle into the interior. And my last topic will be that it's also boring after a while just to take these naked gold nanoparticles or nanorods, but people want to make more elaborate structures. And in Munich there were some new professors coming, young professors actually, who knew how to play with DNA and make origamis out of DNA, which has been first shown, for example, by Rothemund in the US. And we thought about bringing these DNA origamis together with these gold nanoparticles and make some more complicated structures. For example, if you put these such wings on top of a gold nanoparticle, one could think of a kind of a propeller. If we can turn that gold nanoparticle with light, what we can actually. So we were interested in making propellers, and this is done in a way that we take gold nanoparticles and we make like a carpet out of DNA, just by programming the DNA in the right way. I cannot go into details here now. Put some thiol molecules at the end, and then put these DNA carpets onto the gold nanoparticle. And that we can do that is shown down here, that it really works. So these are electron microscope pictures of the gold nanoparticle with these DNA sheets around. And so this is the DNA sheet, which we have to produce first. They can be put down onto a surface and looked at with an electron microscope, and then they have to be attached to the gold nanoparticle. In this case, actually, it was not thiol, but it was just a very simple DNA sequence and the complementary DNA sequence here. And by that, one can put them together and even make one DNA sheet on one gold nanoparticle, or even more. That depends on the concentration of these DNA molecules, and can control in a way how many wings one has onto these gold nanoparticles. So we now want to put them into these tweezers, optical tweezers, into laser focus. And there we have to be careful, because to trap such particles in a laser focus, you need some light power. That means the gold nanoparticle heats up, and the DNA eventually melts, and all these wings break apart. And our first question was, is it possible to find an intensity window where the intensity is high enough to trap the particle, but low enough not to melt the DNA carpet? And we could then take these gold nanoparticles and attach them to the surface, and to check whether they have survived, we looked then afterwards with an electron microscope. So we can put them down like we want, writing DNA or LMU like our university. And the question was, do we have this problem of Icarus from the Greek myth, that too much light burns the wings? And by that we looked on the ground how they arrived, and saw actually there is a light intensity window where they survive that. And so we can use them now in these laser traps for optical manipulation experiments in the future. But we are not so far to show you anything there. So I would like to come to an end and show my group from our last excursion in the Alps. And there you see people like Yek Won-do, is from Korea, doing this DNA origami. And there's Silke Kirchner doing the bacteria detection. And Spas Nedev, he's doing the printing, the optical printing. And Andre Lüttich, he was pushing all these printing experiments, and many others. So this is the end of my talk, the Nano Gold Rush.